Right, good morning everyone. Thanks for coming. Thanks for Bigger also for having me. I'm going to tell you a little bit today about the Fairy Ring project that I've been working on for over 3 years now. It's a project that's been funded by the RNA. Several years ago they decided that we were very short on research into Fairy Ring and there's a lot that we still didn't know. So I'm sure you'll agree it was a worthwhile cause to put some funding into a project purely on Fairy Ring. So this is what we decided to call the project, and it's turned out to be a collaboration between STRI, which was chiefly organised by Dr. Ruth Mann, and Harper Adams University, which is a small agricultural university in Shropshire. So that's my team, and they took me on as a PhD researcher to look at basically the causes and control of fairy rings on golf courses. Now, I've only just finished this. My last data collection was last month, so some of the stuff that I'm going to show you is pretty fresh. There's still a bit of extra data that needs analysing and more things that are going to be put into this, but I'll do my best to show you what I've brought out of it so far, and hopefully there'll be some things of use for you to use on the ground. So, for those of you that don't know, a fairy ring is caused by fungus. So that will have started at a point of origin. So, there we go. That will have started there as a little germinating fungal spore or a fragment of mycelium, and it's essentially pre-programmed to grow outwards. So it's foraging on organic matter in the soil, and it's forever growing outwards, getting older and bigger. And there's a lovely picture, courtesy of Campy, of how it looks, the mycelium in the soil when the turf's removed. And as a lot of you will also probably know, the main problem in dealing with fairy ring as a disease is that it's actually caused by lots of different species. So it's essentially not actually a pathogenic disease anyway. This sort of symptom is just a result of the fungus acting in the soil. It's not actually attacking the grass plant as far as we know. So just a little introduction to the fungal kingdom. One of the main problems with dealing with fairy ring as a disease is that it's diverse. Now the fungal kingdom itself is very diverse. We think up to 1.5 million species, of which we've only identified a tiny fraction of these so far. What we do know about them is that they're some of the smallest and largest organisms on Earth, so from little single-celled yeasts to the biggest organism on the planet, which is a honey fungus that infects a forest in Oregon. So forget what you think about the blue whale being the biggest thing on the planet. It's not, it's a fungus. So they do deserve a bit of respect, and there's a lot that we still don't know about them. So a lot of the diseases that we deal with on turf are classed under the ascomycetes, the spore shooters. The group that we're looking at today is the basidiomycetes. Again, it's a very large and diverse group, over 30,000 species, but all fairy ring fungi are basidiomycetes. So basidiomycetes are basically the group of mushrooms and puffballs, also brackets that you see on trees. Anything that's got a large conspicuous fruiting body is generally a basidiomycete. So the key to tackling fairy ring, the fundamental thing is to know which species you are dealing with. So the aim today really is to introduce you to the ones that occur most commonly on the golf course, of which we can narrow it down to just a handful, and the way that these need handling slightly differently. They occupy different ecological niches, they work in different ways, just like in the animal kingdom, a snake and a badger, for example, are totally different. Just because a mushroom looks like a mushroom, two different mushroom species can be as physiologically different. So I'm going to run through this series of identification tools, ways that we can recognise which species is causing your fairy ring symptoms. So first things first is the type 1, 2 and 3 categorisation system. Now this is a system that was put in place nearly 100 years ago, 1917. The first pioneering paper on fairy rings was by a pair of Americans looking at the Colorado plains, and they categorised fairy rings as type 1, 2 and 3. Type 1 being generally the most severe is when the turf's actually died. That's usually, well, mostly a result of hydrophobicity developing in the soil. One species that we're going to look at in particular, called Marasmius oreades, has surface proteins on its mycelium that are called hydrophobins. Now, they actually repel water. So when that mycelium becomes dense enough in the soil, the whole soil profile can start to repel water, meaning the grass plant isn't getting its moisture, meaning that it dies of drought stress. And there's a couple of other mechanisms included in the type 1 caused by Marasmius oreades, which also emits cyanide into the root zone. So obviously that's toxic to the plant. And another action that that species does is it inhibits the actions of the good bacteria in the soil that convert ammonium to the plant's available form. So again, you get a toxic buildup of ammonium in the root zone as well. So these things combined can lead to type 1 symptoms. Type 2 symptoms, considerably more prolific in the UK, not necessarily as severe, it's more of an aesthetic issue. And this is a result of the fungus feeding in the soil. It's releasing nutrients which are taken up by the plant, and you get this lush growth. So you get a circle or partial circle of darker, greener grass. Type 3 is just the fruiting bodies. So I'm not going to go too much into type 3 because I don't think it's a particular issue for anyone. If you don't like a circle of mushrooms, you either pick them off, mow them over. It's not detrimental to the turf necessarily. But we can get all 3 symptom types together on one ring sometimes. So we use the type 1, 2 and 3 just to break it down into what's actually happening. So that can tell us, it's usually Marasmius that causes type 1 fairy rings. So if you've got turf loss, that's a clue as to what is probably causing it. And I'll show you a bit more about Marasmius now. So this is what I've broken it down to. From that large Basidiomycete kingdom group, which is about 30,000 species, potentially hundreds of species can grow in this ring formation. Some of those will occur in woodlands, some of those occur in agricultural crops, all different types of ecosystems. But I think the ones that we're dealing with on turf, as far as I've seen from visits to golf courses over the last 3 years, I've narrowed it down to these 6 species, or species from about 6 genera. So Marasmius is a little brown mushroom, that's the one I was telling you about that emits cyanide. Agaricus, that one there is the field mushroom, Agaricus campestris. So that's really common, edible and really closely related to the one you buy in the shops. And then the puffballs. Now, these are different puffball genera, but I'm going to lump the puffballs together as one because there is research to show that they do respond quite similarly to treatments. And also, understandably, they're really hard to identify, especially if they're popping up on your greens. They're not reaching mature puffballs, basically, so you're not going to be able to see the characteristics you need to identify them. Often you just get little masses of sort of white blobs pop up that are called primordia. It's not the full mature form. So it's very hard to know which of these puffball species you're dealing with. So I'm going to lump them together and just call those the puffballs. So just to go into a bit more detail, this little brown mushroom, which looks really innocent and delicate, is very, very aggressive. This is what's going to cause type 1 symptoms in probably 90% of cases, I would say. It's really voracious in the laboratory. It grows on anything, it grows over anything. It's a really aggressive species, and it's deep. It's up to 50 centimetres deep into the profile, which is one of the main problems with treating it, is getting to it to a sufficient depth. And if those symptoms become set in enough, then we could have type 1 symptoms like this all year round. And it's your only option if it gets to the point where it's that set in is really going to be to dig it out. So the aim is, as we're going to see, is to manage moisture with these types of rings to stop it getting to a point where it's really hydrophobic and it's only going to be able to be dug out. So this is Agaricus. It's really, really common, especially this year. We had a really dry summer, and as soon as it started to rain, these seemed to pop up all over the place. Type 2 symptoms, it very rarely develops into type 1, and they're not often around for very long either. So although they may be all over the place, they're only going to be there for a month, maybe 2. I have done a bit of work, as we're going to see, on Agaricus, just to see if there were any treatments available. But yeah, they can be very common, but at least they're not here for too long. And puffballs, Don't know if that's come out too well, that photo, but it's the puffballs that cause these watermark type effects that we often see on greens. Puffballs usually occur on greens. If you've got type 2 symptoms on your greens, it is usually going to be one of the puffball species. The good thing is they're shallow foragers, so they're really in the upper layers of the soil, unlike marasmius, which is very deep rooting, which means there is a chance of accessing them to actually get chemicals in if need be. And they're not there all year. They will usually be sort of spring right through to autumn, so they'll generally persist for the whole season. And unlike the marasmius rings, which are very thick set and chunky, these are often very fine lines. You see maybe a thin ribbon crossing a fairway, and say the watermarked effect, that's down to puffballs. So that's fruiting bodies. So as I've just touched upon, ring morphology varies. This is quite a good example. So the one in the foreground, we've got a thin ribbon there. That's a puffball species, and the one behind it, which is the thick brown ring, is caused by marasmius. So just look for these kind of clues to try and help you know what you're dealing with. And then the 3 last questions that we can use to try and identify species is the mycelium visible in the soil? So if you take a soil core out of the ring, if you're on a marasmius ring, the mycelium will usually be thickly visible in the soil. You will see the threads of white mycelium. If you do the same on a puffball ring, you won't usually see the mycelium. Also whereabouts on the golf course. Marasmius prefers fairways. Marasmius likes a nutrient-poor environment, so it will thrive on fairways. It also has its symptoms because it's building up to a dense point, hence why it doesn't tend to occur on greens. Greens are intensively managed, they're aerated, they're irrigated, and they're fertilised, which is why marasmius doesn't like greens, so you'll rarely find marasmius on a green. So that in itself gives us a clue to what we need to do to manage marasmius rings if we've got really nasty ones on the fairways. And also, as I've said again, time of year. So marasmius all year round, puffballs spring through to autumn, and if they just pop up July to September, then that's going to be agaricus, the field mushroom. So good ways to identify which species you have without the fruiting bodies being present. So there are ways to do this. So I'll just run through the sort of things I've been doing during the project. In year 1, any golf course managers here should have had a questionnaire from me which went out to every golf course in the UK and Ireland. Thank you to everyone that did respond, because some of the data I got from that was really helpful. It was quite early on, but it gave me a great background into what the problem was in the UK, how common the different types were, and it gave me a good geographical range of where fairy ring was most of a problem. And some of the things I pulled out from that first year's survey were, as we expected, July and August were the peak symptom times. Type 2s were very common. Also that symptoms were most severe on the links courses. And there was quite a clear divide climatically between the northwest UK, so where it's cooler and wetter, compared to the southeast UK, which had much more severe symptoms, probably climatic things due to the warmth and the dry spells. So I thought this was quite a nice figure, which basically showed each respondent, each golf course that replied to the questionnaire was categorised into one of these colours, which is either that they'd never had fairy ring, they used to have fairy ring but not anymore, they had fairy ring but it isn't a problem, or 3 levels of severe fairy ring incidence. And as you can see there, links golf courses reported twice the occurrence of severe fairy ring compared to parkland and heathland courses. Now, from the research I've done, I think a lot of what this has to do with is that fungi, as far as microorganisms go, are quite large, and they find it very easy to grow and move around sandy soil particles. So fungi like sandy soils. You see heathland golf courses that have got clay soils. Fairy rings are pretty uncommon, and if they are there, they'll grow a lot slower because the large fungi finds it hard to move between the small soil particles of clay. So hence, links courses are going to have more of a problem with fairy ring because they grow more prolifically. So I may have seen some of you in the first year of my study. Quite a few of you were good enough to host a visit from myself. The aim was to get out there and speak to greenkeepers on the ground, get more information about fairy ring, why it's a problem, and also to take samples of fairy rings that I was aiming to bring back to the lab and analyse the DNA able to identify the causative species. Now, unfortunately, I've spent a lot of time on this and it's not come to fruition. And I think the problem is isolating one particular fungal species from soil, which is notoriously difficult because soil is full of microorganisms, as you'll know. So this was one of the gels that I used to display whether you've picked out the DNA you want or not. The two bottom rows there should have looked like the one by the plus sign. So that's my positive control at the top. And that's what I was hoping to see on all my samples, which I didn't really ever see. So unfortunately, that part of the project has not been successful. So I'm sorry to anyone that was waiting for results from soil samples that I've not been able to identify their species. But hopefully some of the other identification tools will give you a clue as to what you're dealing with anyway. So in year 2, I started looking at control. The first control trial I ran was on marasmius rings. Fairy rings are really hard to work on. I had a few cases where I'd start a trial on one and then go back a couple of months later and it's disappeared, or sometimes they'll merge together or move slightly. So it's quite hard to find fairy rings, particularly ones that are big enough to be able to fit a lot of sample plots on. So I found that marasmius in this first instance was quite easy to work with because it's very visible. So I was working on big, mature marasmius rings where I could have lots of plots, and I decided to try the 3 products that were currently marketed for fairy ring control in the UK, which was the fungicide Heritage, a biocontrol from Symbio, Aquacept, and Vitax's clearing, which is just surfactant essentially, all done with and without wetting agent and aeration. On a links golf course and a rugby pitch, and they were assessed before, during and after for turf quality and soil moisture. So unfortunately none of the chemicals had any effect on that trial on turf quality or soil moisture, no improvement whatsoever. What I did see was aerated plots, as you would expect, improved in soil moisture by the end of the trial. But unexpectedly, the wetting agent didn't show any significant effect. And the conclusion we came to from this was that these big, mature marasmius rings are so set in and so dense in mycelium is that the product delivery just isn't getting there. So if the wetting agent's not working, it's no surprise really that maybe the chemicals aren't actually achieving contact sufficiently either. So the conclusion from that was don't bother trying to treat marasmius with chemicals. Basically, you've got to look at breaking down the mycelium to actually to re-wet the ring first. So I built on this a bit. On the next control trial, I decided to work on Agaricus. It's a shallower rooting species. You're more likely to be able to get contact with chemicals. This time I tried Heritage Max and Banamax because, as we'll see shortly, I did some lab work on plates, on Petri dishes with Banamax, and it actually showed to be the most effective at inhibiting fungal growth. I'll show you that in a moment. Potassium carbonate, which was designed as a novel treatment, which again I'll talk to you a bit more about in a moment, and then a treatment of just aeration and wetting agent, and obviously an untreated control. And again, they were tested for turf quality and soil moisture before and after. So there's the results from that one. Again, no significant difference between treatments before and after. So yeah, nothing to report on the control of Agaricus there, I'm afraid. But that's just one trial. So I do think potassium carbonate is worth a mention. I spoke to quite a few guys from New Zealand and from Australia when I was at the ITS conference a couple of years ago, and they were raving about the use of potassium carbonate against fairy rings. So I thought it was worth including in the trial. This is available as a PDF online. This brochure is from the New Zealand Sports Turf Institute, and that's the recommendations for treatment with potassium carbonate. And I'd say just because it wasn't effective on Agaricus rings doesn't mean it might not be effective on other species. So if it is something that you'd like to try, be aware it's alkaline and it's caustic, So you're supposed to add the powder slowly to water, otherwise it can violently boil. So the aim is that it changes the pH quite dramatically to make it unfavourable for the basidiomycete fungi. Beware of scorch if you do decide to use potassium carbonate. I thought I'd watered that patch in really nicely. I hadn't. So yeah, irrigate really well before and after if you do decide to use it. And I just thought this one was worth mentioning because I'm going to come on to the lab trials next, but this is just some of the results from the lab trials. And it just shows that this species in particular, which is one of the puffballs, Hankea rutriformis, the control was on the left there, and on the right you can see that it's taken the whole plate. It grew really quickly over the whole plate there. And that's at a very dilute level of potassium carbonate, but it just shows that this species in particular obviously likes an alkaline environment. So in that case there, which is a lower concentration than you would ever be using in the field, it has made a favourable environment for that species. So each one's got their different niche and different preparations. Okay, so this was the mycelial growth assay, fungicide sensitivity test done in the labs. The one I'm going to show you is just a trial run, which I'm just finishing off at the moment, but this is still to be done. So the experiment proper is about to be set up. It's the last thing I have to do, basically. But I was quite impressed by the results of propiconazole, so I thought it'd be worth showing you. Flutolenil, I know we don't have license for in this country, but it's something that they use, seems to be most often in the States, for fairy ring control. Not something that we will ever have license for amenity, unfortunately, here. But I thought it was worth testing to see if it did work. Pyraclostrobin and potassium carbonate, obviously, to see if anything happened in vitro compared to in the field. So 4 different species tested on. And sorry if this looks a bit messy, this was the trial run. And as you can see, by the rows are the different chemicals. We've got 3 control plates of each at the bottom. The left is Marasmius oreades, that's the small brown aggressive mushroom, and the right is Agaricus campestris, which is the field mushroom. Flutolenil, second row down there, pretty much normal growth, didn't do anything at all in vitro. But the third row down, propiconazole, as you can see from these 2, certainly the best at inhibiting growth across the board, and this is replicated for the other species and the other isolates that were tested as well. So that surprised me because Banamax fairy ring isn't even on the label. So the one that isn't advertised for fairy ring control turned out to be the most effective, so that's just something that really surprised me. So azoxystrobin did do something in some cases, but that's to be done and tweaked and replicated, so hopefully we'll have some good results written up properly from that once it's finished. So moving on from that, I started looking a bit more at the problems with Marasmius oreades and the soil moisture issues with hydrophobicity, and I did a couple of seasons measuring the active zones of fairy rings to see how soil moisture changed as the season progressed in comparison with weather conditions, to try and look at what sort of climatic cycles triggered fairy ring development and the development of hydrophobicity. The main aim of the soil moisture stuff is to be able to say, when your soil moisture falls below such and such a level, hydrophobicity will develop. It was aimed to try and help people to prevent the development of hydrophobicity. But the results have been contrary to the hypothesis, as I'm going to show you. So in the first year, I was just measuring soil moisture. I wasn't measuring hydrophobicity yet at this point. And this is the data that were generated. So the bars at the back there are water input, so that's a combination of accumulated rainfall and irrigation. This is on a links golf course. The orange line is the average set fairy ring soil moisture, and the green is the asymptomatic control. So as you can see, we've got a considerable moisture deficit early on in the year there, which actually came after it was a very dry spring in 2013. So we can basically see from that where water input was lowest, moisture deficit of fairy rings was highest. going to mention? So yeah, I think it's worth looking also in the October. The fairy ring continues to drop, continues to dry out into October, even when the asymptomatic turf is re-wetting. So you can think that the area is getting better when it's actually continuing to fall. So I just think it's important to monitor fairy ring soil moisture. Even early on in the season, it might be drier than you think it is. And monitoring is really important when it comes to stopping the development of hydrophobicity. So I built on this for the second season, 2014, last year, and I added on the water drop penetration time test. So it does exactly what it says on the tin. You put a drop of water on the soil core. If it beads, like that one on the right there, if it takes longer than 5 seconds to completely soak in, it's classed as severe hydrophobicity. If it takes sort of 2 to 3 seconds, then it's a sign that hydrophobicity is starting. And obviously, if it soaks in instantly, it's fine. So I would test that at 3 different levels from each soil core in the same place that I measured the soil moisture content using the Field Scout probe. I also added on a second site this year. I added a parkland golf course as well, so that I could compare the links with the parkland, how the sort of sandy soils compared to parkland soils. And say this is pretty fresh. The first thing to mention was, contrary to the hypothesis, there was no significant link between soil moisture content and the incidence of hydrophobicity. So how I was wanting to say that when soil moisture falls below this level, hydrophobicity occurs, unfortunately, I found that that link wasn't there, which I thought was quite strange. So I had soil cores that were measuring as low as 1–2% soil moisture that weren't hydrophobic. And then again, I had soil cores that were measuring higher. I think the higher was sort of 13% soil moisture that were hydrophobic. So there's something going on there. There's more to hydrophobicity than just soil moisture. There's another mechanism, or maybe a string of mechanisms attached to the development of hydrophobicity. It's not just a result of low soil moisture. So this is, obviously I haven't got the weather data in these yet, but this is just to show you how the parkland compared to the links. Now the links golf course that I showed here is the same set of fairy rings as I showed you on the first year's stuff. As you can see, they're really close right through the year. And I think that was a lot to do with it leading up to a certain big event that occurred there in July. And then I was missing a data point in July, and then after that, the whole area was verticut and top dressed, so I lost one of the rings, and obviously the control and the fairy rings were exactly the same from there on in. So management regimes on the links course in that year have, I think, really impacted on the results. But as we can see, the Parkland golf course, it's a lot wetter overall, but it follows a similar pattern as we saw on the first year with the links. So that's still in quite rudimentary form. That needs data and weather data and things adding to it to try and draw out the relevance. I thought I'd just cover what I did with moisture mapping with you. I wanted to have a look at how soil moisture changed on sort of a 3D level throughout the season. So I was measuring, I just had one marasmius ring that I'd split into a grid of sort of 121 squares, and I was measuring soil moisture at 3 different levels, so at 3.8 centimetres, 7.5 and 12 centimetres deep. So I was measuring that grid at 3 levels. And I did that right through the year from May to November, every 2 weeks. And again, this needs putting against weather data to see how it changes according to moisture and heat. So obviously the darker the green, the wetter it is. And we start in May, really wet, really variable also as well. Move on to June, not much different. But by July, that ring has totally dried out. So it's quite a short period of time for it to develop so dramatically. So that's when it's really becoming active. By August, the lower levels are very dry as well. But by September, it's recovering. And October, it's well on its way to getting back to normal. So I thought that was quite a nice way of displaying it. So from some of that work that I've done, I think most importantly to note is that only a few fairy rings only a few species are responsible for most cases of fairy ring. Moisture deficit is not just something you need to worry about in the summer months. You need to be monitoring for a longer period, thinking about it in the spring. Also to mention, sorry, I forgot to say with the soil moisture stuff, when I was comparing the Parkland and the Links courses, with the Parkland course, my type 1 fairy rings there had turf loss, so they were bare rings, but they weren't hydrophobic. So there's another reason for those to have lost turf. So there is more to type 1 turf loss than just hydrophobicity. So whether that is the result of, as I mentioned before, about there being levels of cyanide or ammonium in there, these still need testing. So yeah, again, there's more to turf loss than hydrophobicity, and as I've said, sandy soils are going to be more affected by hydrophobicity because the fungi are growing better in them. So just to overview how to deal with those 3 main species or groups of fairy ring types. So if you have what you think is a marasmius ring on your course that's a problem to you, the main key is moisture management, what I'm calling poke and soak. So you need to stave off the development of hydrophobicity with intensive aeration, wetting agents, irrigation, fertilisation. So the aim is to stop hydrophobicity, and there's a lot of evidence to show that if you do that intensively, you can stop it progressing and you can counteract and mitigate the symptoms. Whether you'll completely get rid of the fungus, it can be difficult to say. This is a very aggressive, persistent species. But remember, it goes deep. Remember, you need to be getting it in there as deep as you can. Chemicals are unlikely to be effective, so I would say don't waste your money trying to treat marasmius with chemicals. And if it does get to a point where it's that set in, it's going to be very difficult to re-wet, and you'd have to dig it out. It's also worth mentioning that this species is known to antagonise itself. So there's been various cases of people rotivating a whole area, mixing up the marasmius rings. So essentially the individuals are all pitted against each other, and there's sort of fungal warfare, and they wipe each other out. And then if you returf or resod, a few years down the line, it's been shown to stop fairy rings in a marasmius infected area. So the concept of antagonism is something to get thinking about as well, I think. Now, no, I've not said an awful lot about puffballs because I haven't actually done any control trials in the field on puffballs. But there's a chap who has, he's Dr. Lee Miller, he's from the States, and he's actually done a fairy ring PhD just before I started mine. He just looked at putting greens. And as I said before, puffballs are the main species that appear on putting greens, so most of his work was around controlling puffballs. And he did quite a lot on preventative application of fungicides, and I'd refer you to his website. It's great. He's got some lovely stuff on there and presentations about things that they found effective, most of which are chemicals that we don't have available to us here. But he did test azoxystrobin and propiconazole and found them both to be effective on at least 2 of those puffball species, if applied preventatively, either as soon as symptoms appear or the season before to treat for the coming season. So if you've got puffballs, if you've got that watermarked effect, again, think about your cultural controls. Cultural controls are your best weapon, really. But there is a chance of preventative fungicides having an effect here. And lastly, if you think you've got agaricus rings on your course, unfortunately, as I've shown, I don't know any control for that as yet, but at least they're not around for too long. You've also got the option to disguise them with iron or nitrogen if need be, and again, you can do that with the puffballs as well. So yeah, I've not managed to find anything on that one. So the overall conclusion, there is no magic bullet. Fairy ring is not one species. It's not one disease, it's lots of species. So they all need to be treated as separate diseases, if you will, because they all work differently. And as I say, cultural practices, I think, are your best weapon. And a lot of the symptoms that I've seen when I've been out and about on golf courses, I think could probably have been mitigated against or eradicated completely with really sound cultural practices. So I'd really like to acknowledge my sponsors, the R&A, all the supervisory team, especially Ruth, STRI have been fantastic, especially Chris, Bob and Henry, and all my colleagues at Harper. And I'll try and answer any questions that you may have. Okay.
You mentioned at the day after the STRI about trying to take plugs from other ones and trying to get them to fight each other. Did you actually try any of that work at all?
I did a bit in the lab, in Petri dishes, where I pitted marasmius against itself, and there'd be a standoff, and I found the same thing actually to happen with agaricus. So the field mushroom that I showed at the end there, that I said there's no treatment for, in the lab it did actually show that it might have some of these antagonistic characteristics. There's a possibility that that may behave in the same way as marasmius. So yeah, it's something that needs exploring. To replicate it in the field, obviously very difficult and a very invasive procedure to do something like that. But I've spoken to a few golf course managers that have tried it and reported success.
The antagonism dates back to at least the 1960s. And the greenkeepers at that time would take a hand tool that would have like 3 plugs in it, move plugs from one fairy ring area, put them next to the other one, and as most of us know, they never cross over.
Yeah.
They come up and they combat each other.
Yeah.
So I don't know the science behind it, but very effective.
Yeah, well, this is it, and say that's been a concept that's been established for decades, yet still it doesn't seem to be used very much these days. So I don't know what's changed, but I do think that's something that people need to be trying. It's just a really simple thing that you can have a go at, just to swap a few plug cutters around between rings to see if it does eradicate each other, you know. So yeah, it has been shown to work, and I don't know why it's not used more.
Something that may have changed is it was very often in the 60s you would aerify the green and you drag the plugs in.
Yeah.
Today we often remove the plugs and then fill with sand.
Yeah.
So we're not bringing the antagonistic ones from one part of the green to the other.
Of course, yeah, yeah, good idea. Okay.
Would it be important to emphasise with the chemical fungal treatments that you're treating the soil rather than the actual plant? So really we should be emphasising high water volumes?
Yeah.
Yeah.
Yeah, I would say so, yeah.
So we mustn't make the assumption that we just put it on as a leaf application.
Definitely not, no, definitely not. Yeah, we're needing to get contact into the soil, yeah. Anyone else? Right, well, thank you everyone.